I'm David Shih, and I'm with the Pioneers group. So my project is called the Biological Inheritance Expansion Pack, comparing transposable elements and prion-like domains. And so I just wanted to introduce our project premise first and foremost. And our project asks the question, can biological information be stored and passed on in ways other than just DNA sequence? So I'll be looking at the two systems that do this quite differently, prion-like domains, or as we abbreviate them, PRLDSs or PLDSs, and um, they can change how proteins behave and assemble, and transposable elements, or TEs, which can move or copy DNA within the genome. And the goal is to ask whether these two systems might perform similar functions at different biological levels, and what that can tell us about biological memory, evolution, and the generation of new phenotypic traits. And so, one of the clearest examples of information being carried by a protein comes from prions. And so we have diseases like mad cow disease and Creutzfeldt-Jakob disease, which helped establish the idea that a protein could adopt an abnormal shape and then cause other copies of that protein to adopt the same shape. And that was important because the information was being passed through protein conformation rather than a change in DNA. And later, similar self-propagating protein states were discovered in yeast, including Cy+ and URE3. And many of these yeast prions contain particular regions called prion-forming domains. And their sequence features eventually led to tools like plaque, which can identify proteins with similar prion-like domains. But there's an important distinction where a prion-like domain is not automatically a prion, and many prion-like domain containing proteins never behave as self-propagating prions. Instead, they can help proteins assemble, form condensates, and regulate RNA or respond to stress. And so the broader idea is that protein state itself can sometimes carry biologically meaningful information. On the other side, we have transposable elements, which are DNA sequence that can move, or historically were able to move to new places in the genome. And there are two major types. We have DNA transposons, which generally use a cut and paste mechanism, and we have retrotransposons, which make an RNA copy first, then copy that information back into DNA somewhere else in the genome. And TEs were originally thought of as mostly disruptive DNA because their movement can cause mutations or interfere with genes. But over evolutionary time, some TE sequences have been repurposed by the host, and they can become promoters, enhancers, and even contribute to coding functions. So although TEs can be disruptive, they can also become sources of biological innovation. And so we want to be clear that we're not saying that prion-like domains and TEs use the same mechanism. It's clear that they don't. Prion-like domains act through things like protein shape, assembly, and interactions, and TEs act through DNA sequence. But if we zoom out and um, we can see that sometimes they both create states that persist, generate variation, and alter larger biological systems. And so my comparison is about more what they do rather than how they do it. So we're comparing them as functional analogs. And so I do this by using four functional axes. The first is information transmission, which refers to, can the state or element be passed on or reproduced? The second axis is biological memory, which asks, does its effect last after the original trigger is gone? And third is reversibility, which asks, can that state be removed or returned toward baseline? And the fourth is modular innovation, which asks, can that system be reused into a larger biological function. And so my premise is how prion-like domains and TEs perform comparable information processing functions at different biological layers, and what this might reveal about early evolution and biological memory. And so between prion-like domains and TEs, they have different substrates, but these are different substrates with comparable information functions. So for prion-like domains, the information is associated with the state of the protein and its shape. But for TEs, the information is stored in things like the DNA sequence and genomic position, and they also spread differently. So protein states can sometimes influence other proteins or persist through assemblies, while TEs spread by copying, inserting, and being inherited as DNA. So even though the physical carriers are completely different, both show that biologically important information can exist in more than one type of molecular form. And this is where the idea of biological memory becomes useful. So for prion-like domains, that memory can exist as a protein state, and for TEs, it can exist as a DNA insertion or another genomic change. And the main difference being durability, where protein states can persist, but they also disappear through things like dilution or degradation, where a TE insertion can remain in the genome for much longer, and this can persist even across evolutionary time. So one way to think about this is that prion-like domain-associated states provide a more flexible form of memory, while TE insertions provide a more durable version of this. And so both systems can also be thought of as modular, meaning they can be reused in new biological contexts. And a prion-like domain can change how a protein assembles or which proteins and RNAs it interacts with. And a TE can introduce regulatory DNA like a promoter or enhancer into a new genomic location. So over time, either type of component can become incorporated into normal cellular regulation. So evolution does not always have to invent something completely new, and sometimes it can just reuse an existing place in a new context, which is what we call co-option. And we also have reversibility, which is one of the clearest differences I saw between the two systems. The prion-like domain associated states can be reversed because proteins can disassemble, be degraded, whereas TE activity can be turned back off through silencing. But once a TE has inserted into the genome, that DNA change is much harder to remove. So some DNA transposons can excise, but even when they do, they leave a sequence footprint. So the important distinction is not whether it's active or inactive, dormant or not. It's how easy it is to erase the information that was created. So this is why I think the two systems represent a complementary type relationship where one tends to be more flexible while the other can create a more durable genomic record. And so going over my methods, I built a preliminary data set from the literature, and each named prion-like domain or TE system was treated as one example. So even if several papers studied a single prion-like domain or transposable element system, it wouldn't be so heavily counted in my criteria. And then I scored each system from 0 to 3 across the four dimensions that I described earlier: transmission, memory, reversibility, and modular innovation. And then I compared each dimension separately. So a score of 3 in transmission does not necessarily mean the same thing as a 3 in reversibility. And the goal here wasn't to create a final answer or conclusion, but to identify which parallels looked strongest and which ones deserved further study. And so, looking at my heat map, if you look on the left here, I have all of the prion-like domain and transposable element systems. And this summarizes the literature coding. And the main thing to look for is the overall pattern. And we see transmission and biological memory were generally high in both selected groups. And the visual difference seemed to be reversibility, where the prion-like domain associated systems tended to score higher. And modular innovation was also pretty common in both groups. So this gave me an initial picture of where the two groups seemed similar and where they began to differ. And I also did a Gower distance to summarize how different two systems are across all four dimensions. Then I used a PCOA to turn those distances into this two-dimensional plot. And the easiest way to read this is that the points that are closer together have more similar profiles. So the prion-like domain and TE systems showed some separation but not complete separation. So they're not functionally identical, but not completely unrelated either. And in this pilot, the system type, whether it was prion-like domains or TE systems, explained about 22% of the variation. And I treat this as exploratory, especially because across these four dimensions, they represent different biological properties. Transmission and biological memory both had median scores of 3 in the two selected groups. So those looked broadly similar. That means the mechanisms were demonstrable in literature, and the clearest difference was again reversibility. And the prion-like domain group had a median of 2 compared with 0 for the TE group, with a permutation p-value of about 0.0025. Modular innovation was high in both groups, but there wasn't a clear statistical difference. So the strongest quantitative difference in this pilot was how reversible the information-bearing states were. And going on to my result patterns, three things stand out. Transmission and persistent effects were common in both, and both groups shared the ability to contribute to larger biological systems. And third was reversibility was the clearest difference. And this is also corroborated by literature, um, corroborated like by literature and non-prion-like domain systems such as FUS and HNRNPA1 can form dynamic and reversible condensates. By contrast, once a TE successfully inserts into the DNA, and that genomic change is generally much more persistent. And so for my limitations, I first selected these systems from literature through a systematic review. So the percentages that I mentioned earlier should not be treated as estimates for all prion-like domains and TEs. And the prion-like domain side of the original data set also contained too many true prions. And third, I did the scoring myself, so there's some arbitrariness to like what you saw in the scoring criteria of the 0 to 3 grading of each prion-like domain and TE system. And so for my conclusion, um, the main implication is that early biological systems have used multiple forms of templating and inheritance rather than relying on one information substrate alone. So this is why I describe it as complementary. And um, this suggests that they're both biologically viable strategies, which is called evolutionary pluralism prior to genetic takeover, where the central dogma that we're familiar with predominates. So I just want to thank the pioneers and my project mentor and Blue Marble Space in general. Thank you guys for listening.